Assalamu alaikum. Today I am going to discuss about double beam spectrophotometer. Last time I have already discussed about single beam spectrophotometer. Now let's see what is double beam spectrophotometer. What is double beam spectrophotometer? First point is a double beam instrument divides the radiation into two beams which are passed through separate cells. This point explains that radiation coming from the source is split into two beams, one passed through the reference or blank and other passed through the sample. The second point is in double beam instrument, one of the two cells, one is filled with blank solution and the second cell is filled with the sample. It means one cuvette is for blank and other cuvette is for sample. The third point is, since the readout from the instrument is the difference between the amount of light absorbed in the two cells, the absorption in the sample is automatically corrected for absorption occurring in the blank and for changes in intensity of the incident radiation with wavelength. What does it mean? It means radiation passed from both cell, blank and sample. Blank absorbed radiation and sample also absorbed radiation coming from the source, but instrument automatically subtracted blank reading from the sample reading and we only get the sample reading. As we know, blank only contain matrix while sample has analyte and matrix as well. By the instrument nullified the matrix effect in the sample. On screen you can see the schematic diagram, light source, monochromator, slit, rotating disc mirror, sample cell, reference cell, detector, computer. Radiation coming from the source, then monochromator split the radiation into its component. From the slit only that wavelength pass which we want to use for analysis. One pass from the sample and the other pass from the reference and then detect by the detector and then read out by the recorder. Components of the instrument: source, monochromator, wavelength selector, cell or cuvette, and the last one is detector. Let's discuss the components in detail one by one. First one is source. So different types of source used for different electromagnetic radiation region. First one is tungsten filament lamp. The source of visible radiation is usually a tungsten filament lamp. It means for the visible region we use this lamp, which provides continuous radiation from about 320 to 2500 nanometer. This is the range of this instrument. This is the range of this source. Now the second source is hydrogen lamp, deuterium lamp or xenon discharge lamp. In the ultraviolet region, the radiation source is often a hydrogen lamp, deuterium lamp or a xenon discharge lamp. It means this region we used these lamps. The hydrogen and deuterium lamps provided continuous radiation from about 180 to 350 nanometer. This is the range of these lamps. The xenon discharge lamp provide UV radiation which is usually more intense but less stable as a function of wavelength than that from the hydrogen and deuterium lamp. Here you can see the picture of the deuterium and xenon discharge lamp. Now the second component of the instrument is wavelength selector, a device which narrow the bands of electromagnetic radiation coming from the radiation source to the relatively narrow band which is used. This point means a device which narrow the bands of coming radiation from the source. There are two major ways to limit the wavelength band. First one is filter and the second one is monochromator. Now first one is filter. In some instruments filter is placed in the path of the radiation. It means we used optical filter in the instrument for wavelength selection. only specific or particular wavelength pass through the filter. Filter prevent passage of radiation at all wavelengths except in a fixed wavelength region that is typically 20 to 50 nanometer wide. Filter instruments are relatively inexpensive. The disadvantage of this instrument, the operator to change the filter whenever the wavelength band to be used in this study is changed. In our lab, we used photometer in which we change the optical filters. Now, monochromator. Monochromator limits the wavelength band passing through the sample. It means particular wavelength passing through the sample. The second point, a monochromator usually consists of entrance and exit slits surrounding either a prism or a diffraction grating, which is used to separate the radiation according to its wavelength. Basically, these points show the composition of monochromator. For this understanding, we should know that what is prism. Prism that separates light into a spectrum of colors. As we know, prism operate by refraction. What is refraction? Refraction is the bending of a wave which travel from one medium to another. And then what is diffraction grating? What is grating? A plate of glass with very close parallel lines producing a spectrum by diffraction. What is diffraction? Diffraction is the bending of light as it passes around the edge of an object or obstacle. Diffraction grating operate by diffraction. The third point is certain wavelengths focused on the exit slit of the monochromator are allowed to pass through the device and into the cell. It means as the slit width is increased, more radiation passes through monochromators and wavelength bandwidth also increases. The slit width is normally adjusted, which will still allow enough radiation to pass through the monochromator to provide for accurate measurements by the detector. The operator usually has control of the width of the exit slit. Here you can see the composition of the monochromator. Entrance slit, dispersive element, maybe prism or diffraction grating, exit slit, and then we get monochromatic light. As the slit width increases, so many wavelengths we get, and as the slit width minimizes, so we get only monochromatic light. And this slit width controlled by the knob. On screen you can see the schematic diagram. Here is source, entrance slit, dispersion device, exit slit, sample, detector. From the source, radiation coming and pass through the entrance slits and then dispersed by the dispersive elements, maybe prism or diffraction grating splits into its component, and then particular light pass through the exit slit and then pass through the sample and then reached by the detector and then read out by the recorder. So this is all about the schematic diagram. The wavelength of the radiation to be used in this study can be adjusted by turning a knob on the instrument. Here you can see the picture of the instrument. By the help of this knob, we can adjust. The wavelength band passed through the monochromator is usually narrower than that passed through a filter and can be narrow as 0.1 nanometer. Now the third component of this instrument is cells or cuvette. What is cell? The solution is held in place in the path of the electromagnetic radiation in a container known as cell or cuvette. Now the second point is the substance from which the cell walls are made and through which the radiation passes must be transparent to the radiation. Why must be transparent to the radiation? Because if we used colored cuvette, so it absorbs that radiation which we used for analysis, and definitely our analysis become incorrect. So that's why the reason is that we used transparent cuvettes, because all the radiation must be passed from the cuvette. In the visible region, the cell can be made of glass. For the visible region, cuvette is made of glass. In the ultraviolet region is usually made of quartz or fused silica. In the UV region, cuvette is made of quartz or fused silica. Here you can see the cuvettes. First one is made of glass and used for visible region, and the second one is made of quartz and used for the UV region because it passes the UV radiations from it. Later I will discuss the last component of this instrument, which is detector. Thank you for listening. Please let me know if you have any queries about this lecture.